this is what your tree should look kind of like. And your outbreak strains are down here, which is good. And they're all closely related, which is what you would expect in an outbreak. If they're all over the place and amongst other ones that aren't from the same ward, it's probably not really an outbreak. If we look here, so this is from the same study. So each of these rows represents a patient, a baby, I think, and most, well, the ones with the white background are babies, so all of these ones here. And this is at certain time points when they were swabbed to see if they have MRSA. And then if it's got a dot in the middle, it means they were swabbed and that MRSA was sequenced. And so the one we're interested in is 6C. And your suggestion for time is very good, but it was taken the same time as some of the other strains. So it doesn't seem to be any earlier. And in fact, these ones were sequenced as well in there. Well, they're over there somewhere. Yeah, so it's not that. And actually, the people that did this study, they looked at it and they found that this particular strain, because it's quite strange that it would be quite separate from the other ones, they found that it's something called a hypermutator. And it's just one of these things that sometimes some bacteria, the genes are slightly different and it causes them to mutate at a higher rate, which can be an advantage because it means that you can adapt to your environment more quickly, but it can be a disadvantage as well because you could get bad mutations, like erroneous mutations. So interestingly, one of the other things that they found in this study is, I was saying about with the whole genome sequencing, that you align everything to a reference genome. If you think about it, you're not going to get all of the information that way. So you can look at what's common to everybody, but if some of the strains that you've sequenced, if they have extra DNA that's not in the reference, then there's nothing for it to align to, if that makes sense. So we have what we call the core genome, which is something that's common to everything, and then we have the accessory genome, and that's that part that I was talking about, horizontal gene transfer, where bacteria can exchange genes with one another. And they tend to exchange genes that are useful. So if you're a pathogenic bacteria, the kind of things that might be useful for you would be virulence genes or antibiotic resistance. And so on top of looking at the core genome and seeing the relatedness and determining that there is an outbreak, a true outbreak in this hospital, they also looked at antibiotic resistance genes. And they found that these outbreak strains here, in fact, is that the next part in your thing? The outbreak strains have different antibiotic resistance genes compared to the non-outbreak strains. And in fact, they have more. So these Rs confer resistance, and so the blank parts mean that there's no resistance. So you can see another thing to confirm that they're from the same outbreak. They have the same antibiotic resistance profile, we call this. These are the outbreak strains. And they have more antibiotic resistance genes, which might explain why they were quite successful in the outbreak. So I think we'll stop there. There is a final exercise, but that's like a extra credit exercise, if you like. And that's for looking at whole genome data and actually visualizing it. So if you want to look at the assembly of the data that you've made, you can actually look at it and compare them. The software that you need is something called Brig. It's all in there. You should be able to download it. It's free to download. So if you have any questions, please contact me. If you have a go at the final exercise and you get stuck or anything like that, please contact me. I sit at a computer all day for my job, so I'm very readily available and I'm more than happy to help you.